Today we learn about communicable disease or the disease caused by pathogens. So what defines a pathogen? Pathogens are those organisms which will cause a particular disease in an organism. For example, like viruses, bacteria, helminths. So these are the pathogens or the germs which can cause any type of diseases in an organism. So, first of all, here remember about the germ theory of disease or the germ-line theory of disease, which was mainly postulated by Louis Pasteur and Robert Koch in the year, we can say 1866 to 1885. Most important thing over here is that Robert Koch demonstrated the presence of rod-shaped bacteria. Remember, the presence of rod-shaped bacteria called Bacillus anthracis in the blood of the cattle which are suffering from a disease called anthrax. So the Robert Koch was the one who discovered this rod-shaped bacteria that causes a disease anthrax. The scientific name of that rod-shaped bacteria is Bacillus anthracis. So therefore, Robert Koch in 1884 he identified 4 conditions that mainly attributes to a disease which is caused by a particular pathogens. Alright, so he identified the 4 conditions and attributed that these particular conditions will lead to a particular disease which is caused by particular pathogens. And later on, these 4 attributes which was discovered by Koch was later known as Koch postulates. So now we discuss about the Koch postulates or the Koch attributes for pathogenic diseases. So what are the 4 attributes? Which was mainly been discovered by the Koch. So first one, we can show here. The disease-causing organisms or the pathogen must be found in all the animals that have the disease. That means we have learned that a particular pathogen will cause a particular disease. So therefore, if an animal is suffering from a particular disease, if an animal is suffering from a particular disease, then the respective pathogen that causes the disease must be present in that particular animal. So first postulates, the disease-causing organism must be found in all the animals that have the disease. So any animal which is suffering from a particular disease, so therefore the respective pathogen must be present in that animal. Number 2 attributes, the pathogen must be isolated from the diseased animal and grown as a pure culture on an artificial culture medium, mostly agar. So number 2, so if an animal is suffering from a disease, the pathogen is present over there. Number 2 conditions, now we have to isolate. We have to take out the pathogen from the animal suffering from a disease. And that pathogen must be cultured in a particular culture medium containing mostly agar. Are you getting it? So first one, any animal which is suffering from a disease, it must contain a pathogen. Number 2, we have to isolate that pathogen from that diseased animal. And we have to grow that pathogen in a culture medium, in an artificially cultured medium. Clear? Number 3, inoculation of pathogen from culture into the experimental or the non-diseased susceptible animal must possess characteristic symptoms of the disease. What do you mean by this? So those pathogens which has been cultured in the artificial culture medium now must be inoculated, must be introduced into the experimental animal. That animal must be free from disease. Clear? So that pathogen which is grown in the culture medium now must be inoculated into the experimental animal, and once it is introduced into the experimental animal, that pathogen must be able to produce the characteristic symptoms of the disease. Right? So number 4, the same pathogen must be recovered from the inoculated diseased animal. That means the pathogen which is grown in the culture medium and which was introduced and which shows the characteristic symptoms of the disease. Right? Now that same particular pathogen, number 4, the same particular pathogen must be recovered from this animal also. So these are the 4 attributes, or we can say the goals, postulates for pathogenic disease. What are the conditions? If any animal is suffering from a disease, then respective pathogens will be present in that animal. Number 2, isolate that pathogen from the animal, and we have to grow that pathogen in an artificial culture medium. Number 3, that cultured pathogen must be introduced into the experimental animal, and that animal must show a characteristic symptom of the disease. And lastly, number 4, the same type of disease, alright, that is caused by the same pathogen must be recovered from the same animal. So this is the goal, postulates for pathogenic disease. And what are pathogens? Those organisms which will cause a particular disease in an organism. So now we'll talk the next one, reservoir of infection. Okay, next one, reservoir of infection. That means the particular place or the source where the pathogens are present, but they are not able to cause the disease. It's called the reservoir of infection, where the pathogens are present, but those pathogens will not be able to produce the characteristic symptoms of the disease. So let's talk about this one. Every disease producing organism has a reservoir of infection. So each and every pathogen that causes a particular disease has got a reservoir of infection where it normally lives, but does not produce or cause a disease. So the reservoir can be soil, water, air, animals, or other human beings. That are the carriers. So the soil, water, air, or any other animals, they can act as a reservoir of infections where they can live, but they are not able to cause the disease. So let's talk about the carrier or the vector, which are those organisms that will transmit the pathogens from one place to another place. So we can say that from an infected individual to a healthy individual. So they are simply called carrier or vector. You know the difference between carrier and vector. So they will mainly help in the transfer of pathogens from an infected individual to a healthy individual. Number 2, soil, which also acts as the reservoir for Clostridium tetani, air, with reservoir for bacteria, cyst of tuberculosis and diphtheria, virus of measles and mumps and other disease-causing bacteria. Food and water are also reservoir of bacterial cysts of cholera, typhoid, Entamoeba histolytica, helminths like Taenia, Ascaris and Cochlearia. So these are the reservoir of infections which act as the reservoir for the pathogens, where the pathogens can live freely but they are not able to cause the disease. Now next one, what are the modes of transmission of diseases? How the pathogens are able to transfer from an infected individual to the healthy individual. So now we learn about the mode of transmission of diseases. We have got simply 2 modes, direct and indirect. Okay, we have got direct transmission and indirect transmission of pathogens which will cause a particular disease. So first one, we learn about the direct transmission of pathogens that cause a particular disease. So under direct transmissions, here the pathogens will be transmitted from an infected individual to a healthy individual without any intermediate agent. So it works in the following way: by direct contact with an infected person. Okay, so whenever an individual is infected and when the healthy individual comes in contact with that individual, then the disease will be transmitted. This is called direct contact. So this is like contagious disease which can be transmitted easily, like chickenpox, smallpox, okay, these foods, measles, leprosy, ringworm, gonorrhea, syphilis. These are the example of disease which can be transmitted by direct contact. It can be spread by the physical contact between the infected person and healthy person. The mouth amoeba, Entamoeba gingivalis, is transmitted by direct mouth-to-mouth kissing. So the example of direct contact with infected person. Number 2, droplet infections. What are the examples? Like diphtheria, influenza, common cold, tuberculosis, pneumonia, mumps, whooping cough. They are mainly transmitted by the help of droplet infections, where the germs are present in tiny droplets of mucus and they are mainly released during sneezing, coughing, spitting, or even talking with the infected persons. Number 3. Contact with the soil, because soil is also reservoir of many harmful pathogens. So mainly in the soil we will find the bacterial cyst or tetanus, alright, which mainly enter the human body from the soil through the injuries. Animal bites also, mainly the rabies virus, alright, it can enter into the body by the bite of a dog or any other, let's say pigs or the cattles. And even last we can say over here, from the mother to the fetus also, most of the disease can be transmitted through the placenta. So through the placenta also, the disease can be transferred directly from the mother to the developing fetus. So these are the different modes of transmission of disease under the direct mode of contact. Next one, indirect transmission. So let's figure out. In this case, pathogens are transmitted from the reservoir of infection to a healthy person. In case of direct transfer, there was no intermediate agent for the transfer of pathogens. But over here in indirect transmission, the pathogens or the disease will be transmitted from a reservoir of infection to a healthy person. And you know what is reservoir of infection now? Here. Now, it also requires an intermediate agent. Here, so let's see now the first one, what is the agent? Vector. So vector, the insects transmit disease in the following way. So first one is talking about the hospitalized carrier. Remember, the parasites or the pathogens are not dependent to complete its life cycle on the body of a carrier, but they are dependent on the vector to complete half of its life cycle. So hospitalized carrier which carry the pathogens for cholera, typhoid, dysentery, and tuberculosis. Okay, and remember the name of that particular agent that causes tuberculosis is mycobacteria. Clear? Bacteria. Now, these particular pathogens will be transmitted by the help of hospitalized. So, those particular pathogens will mainly attach to the spiny leg of the hospitalized or the mouth parts from faces and sputum to our genitals. So, hospitalized will carry the pathogens from one place to another place. And mainly when those foods will be contaminated by those pathogens, and when healthy individuals will mainly take those foods, that individuals will be infected by these particular diseases. Now when this contaminated food is taken, it causes infection. Housefly also carries microbes responsible for ophthalmia and conjunctivitis from eye to eye. You know what conjunctivitis? Cockroaches and crickets also contaminate the food with these germs. Simple. Housefly is a carrier that will cause the pathogen for diseases like cholera, typhoid, dysentery. So it will carry the germs from one place to another place. And if the food will be contaminated by those germs, and even healthy individuals will take those food, then that individuals will be infected by those pathogens. Clear? Next one. Certain blood-sucking insects transmit disease-causing organisms in the host body. Human body uses scripts, typhus, rat, flea, guinea pig, chestnut fly, African sleeping sickness, sand fly causes kala, oriental sore, Aedes mosquito, yellow fever, Culex mosquito, filariasis, Anopheles mosquito, malaria, and ticks transmit Rocky Mountain scarlet fever. So these are the diseases that are caused by the blood-sucking insects. May you remember? Anopheles mosquito, they are mainly the vector. So by the help of this, the disease can be transmitted through an indirect mode of transmission of diseases. Next one, vector. Pathogens of cholera, dysentery, typhoid, hepatitis B are transmitted by the diseases like food and water. So mainly those pathogens which cause hepatitis, typhoid, dysentery, they are mainly transmitted by the help of food and the water. So if the food and water are contaminated by those pathogens, remember carefully, if the food and water are contaminated by those pathogens that cause this particular disease, then individuals who are taking up those food and water, then that individual will be infected by these diseases. Next one, airborne. That means pathogens are present in the air. In this case, pathogens are spread by wind, aerosol spray, and dust. Format 1.
Many pathogens are transmitted through the contaminated articles such as towels, handkerchief, crockery, toys. So that means whenever these particular articles will be used by healthy individuals, then yes, he might be infected by those pathogens if it is being used by the infected individuals. Or it will hence germs of disease like dysentery or hemobiasis, alright, ascariasis, enterobiasis are spread by taking food with unclean hands. If our hand is contaminated by those pathogens which causes ascariasis, which causes enterobiasis, which causes dysentery, then we will also suffer from the same disease. If we used to take the food by the unclean hands. So today we have just simply learned about the disease causing pathogens, what are pathogens. We have learned about the cause postulates. We have learned about the mode of transmission of diseases. On that, we learned about direct transmission and indirect transmission. The rest of the topic we will cover it in the next class. Till then, stay safe, study. Thank you.